going to get a little bit more on echinoderms, on echinoids. And first, I would like to thank the group of people that have been working on this project. Jeff Thompson, who is here, a grad student in my lab who's done most of the work I'm going to talk about, but also Liz Petzios, also in my lab, and then a recent graduate of Eric Davidson's lab. Eric Erkenbrack is now a postdoc at Yale. We were part of a larger effort coordinated with Eric Davidson's lab to try and look at gene regulatory networks and to see how they developed over evolutionary time. And indeed, that is sort of the thrust or theme of my talk, which follows up on what we heard last night from Neil and from Allison and from Veronica, is that we're now beginning to enter a phase where we can look in evolutionary time, which we might also call geological time. It's just not historical time, but we also call deep time, tens of millions, hundreds of millions of years, and understand some features of regulatory networks and when they might have evolved. And Veronica had some interesting results on that, and others have also talked about this. I think we're really at the forefront of beginning to know this level of detail. And as you know, geological time is known from studying this very interesting instrument called the Sedimentary Rock Record. We've heard a little bit about that. Here's the Grand Canyon. And if you study fossils, all of your data has turned to stone. So it turns out that a lot of us are people that pass through geological training. And we know, as Allison and Neil pointed out, that there is lots of information through this deep geological time about evolution, and that's why it's an important component from studying fossils. This is a phylogenetic tree for the birds. We have a zebrafinch here, and we know that birds evolved in the Mesozoic, but that the big mass extinction at the end of the Cretaceous, most of them went extinct. A few passed through to give us our modern birds. If all we had was modern birds, we wouldn't know about the sort of rich history, rich evolutionary history of birds. So the fossil record brings a lot of really interesting components that we find fascinating. We heard about these Cambrian organisms earlier on as another example. But this evolutionary time, I find, is a thing that's hard to wrap my head around, and I've been doing this for 40 years. And it's a hard thing to teach to people, mainly because I think we as humans have a real trouble with big numbers, and that's why Las Vegas exists. So if I say, oh, 100 million years ago, 200 million years ago, we all kind of go, uh-huh. But still, it's really hard to understand how much time there has been for evolution to occur. And we have seen just the extraordinary complexity when we get down to the molecular level. And I think that we can reconcile ourselves with that when we throw this concept of deep time into it. And yet, what's really fascinating that we're beginning to learn is that a lot of these genetic apparatus is fairly conserved. It maybe evolved a long time ago. And a lot of it is functional. I guess what works, sticks around. So this side, this is a, I think you're familiar with the geological time scale. I like this version by Ray Troll, sort of a pop artist, but he's very interested in fossils and things. We have the sort of very fossiliferous part of the geological record. Allison was talking about the Cambrian explosion down here. 542 million years ago is where the Cambrian begins, and that's where we have a rich fossil record. The bird and other theropod dinosaur extinction happens here, and if we're talking about echinoderms, this big extinction happens and is also important in their evolutionary history. So lots of time. We have studied a lot of fossils. But now it's time to try and link together what many of you here do, what you're experimental scientists looking at the molecular biology, at the regulatory networks, at the coding genes, at all of the really stuff that makes animals, and try to throw this component of deep time. And this concept of paleogenomics, it's a field that has a variety of different meanings. It could mean, if you're doing paleogenomics, that you're looking at mastodon DNA. But in 2006, in a paper that I, with Andy Cameron, who's here, and Eric Davidson and Kevin Peterson, we proposed that we could use, that as paleogenomics, we would add the component of deep time to genomics. And we use an example, one of our favorite groups of organisms, the echinoderms. Here's a tree for both the modern and now extinct groups of echinoderms. And we noted that the character that makes you an echinoderm is the skeletal material called stereome, which we see here, some echinoderm modern and ancient skeletal material. And this first is found in the fossil record in the early Cambrian, and that it is sort of iconic for echinoderms like bone is for vertebrates. It has a very characteristic structure. And we linked this with the work being done on the purple sea urchin and the skeletonization genes and the regulatory network for that, where we basically said we think we can infer that this set of processes here, the regulatory apparatus, the biomineralization genes, and other features, probably first evolved in the Cambrian and was making stereome, and that variants of this have existed ever since then. So this is a pretty conservative process, and that stereome is still being made today. So maybe what works, works, and you stick with it. Obviously, there have been variants on this. Okay, so let's talk about a wonderful group of organisms, echinoids, and see if we can do some more with this sort of approach. Sea urchins have got, we have the purple sea urchin here, Strongylus centrotus, but there's lots of other morphologies. It's really a highly diverse in modern ecosystems. Some of the ones that are most fascinating are things like sand dollars and heart urchins. And as we've said before, when we do some molecular phylogenetics, we see that for the modern echinoderms, here are the sea urchins along with the other crown groups, the sea cucumbers, crinoids, ophiuroids, and sea stars. Usually when we look at this variety of diversity, we can sort of not really phylogenetically, but just lump the morphology into regular echinoids, which are epifaunal and move in any direction, usually scraping the substrate for food. And we see quite a range here in morphology of regular echinoids. And as I've already noted, there are the wonderful irregular echinoids, which have adapted to living, sort of burrowing a little bit to burying a lot into sediment. So we have sand dollars and heart urchins, and there's quite a marvelous sort of ecological and taxonomic diversity of this group. The echinoid fossil record is pretty good if you want to study their evolution. Here we have Bothrioceteris, an Ordovician, earliest known echinoid. Through the rest of the Paleozoic, we have pretty good, these are partial specimens, not complete whole specimens, because in the Paleozoic, things tend to fall apart, but we have a pretty good record to the rest of the Paleozoic. In the Mesozoic, we start to get a real diversity of these irregulars, and they have more sutured skeletons, and we get more complete specimens. But it's a long record, 460 million years. In the post-Paleozoic, we have two groups, subclasses of echinoids. And these are the ones that we find today also. So after the end Permian mass extinction, that big mass extinction we saw earlier on in the time chart, we have the sideroids, sometimes called the pencil sea urchins because of their spines, their thick spines. They're all regular echinoids, and they haven't really diversified that very much. So they sort of stick to the same game plan throughout the post-Paleozoic, the last 250 million years. But then the other subclass are the euechinoids, and as you can see, there's been widespread diversification. Many of them are regular, but also a lot of this diversification is due to the evolution of the irregular shape, morphology, and the ecology that goes with that. So let's focus in now on sideroids and euechinoids, since they're the two crown groups that are around with us. And we know that the most studied is the purple sea urchin here, and then we have Eusideris, which is a sideroid. And perhaps the most recent publication to come out of the Davidson lab is one last summer with Eric Erkenbrack, and where the differences in the gene regulatory networks between sideroids and euechinoids were highlighted, although we already knew that these kinds of differences were forthcoming. Okay, so let's talk about a classic feature of the echinoid regulatory network, first discovered by Paola Oliveri, when she was in Eric's lab, and she's here. And so if you want to learn more about it, you can talk to Paola. And this is the famous double negative gate in early development of euechinoids. And this is where we have the gene PMAR, and it's expressed for those micromeres that Veronica showed us in early development, as we can see here in these in situs. But for the rest of the embryo, PMAR is not expressed, as you can see. And PMAR is a repressor for Hess-C, so that in the micromeres, we don't see Hess-C expressed, whereas since PMAR is not expressed for the rest of the embryo, Hess-C is expressed at this stage of development for the rest of the embryo. The double repression mechanism, or the double negative gate, is that because of the repressor and repressor, we get then, with Hess-C being repressed, it represses these downstream genes shown here, and they are expressed in the micromere lineage, whereas the rest of the embryo, they're not, because Hess-C is not repressed. So this is a classic feature of the gene regulatory network of echinoids, and found in euechinoids, I should say. And if we put it into the larger context, here is the double negative gate mechanism, and the downstream genes are the ones that we saw earlier for stereome formation. So it is part of the skeletonization gene regulatory network, these regulatory genes. So it's part of that bigger picture of how do we make skeleton and stereome. Okay. Recent work in this paper that I showed you before by Erkenbrack and Davidson has shown that Hess-C and those downstream genes overlap in expression, so that in sideroids, I should say this is in sideroids and Eusideris, we don't see the double negative gate operating. And this is work that has also been shown for a different sideroid by Yamazaki and others. And they've been very active studying this feature also. So sideroids actually, in making the larval skeleton, do it at another place and another time in development from what we see with euechinoids. And so by not having the double negative gate, there is a difference in how the whole early skeleton is set up in the larvae, but it's still using the same biomineralization genes that we talked about earlier on. So here we have a gene regulatory network, system, a piece of it. And the question is, when do, and it's different between these two subclasses, when do these changes occur? And we can go to the fossil record where Jeff and others have published a recent paper on a sideroid specimen, shown a piece of it here, found in some cabinets at the Smithsonian that has an age of approximately 268 million years ago. It seems to be the oldest sideroid currently known. And due to cladistic logic, you infer then that euechinoids were also around then. These are, of course, specimens of adults and not embryos or larvae. There is no embryo or larval fossil record for echinoids. But so we have adults here and definitely a sideroid, and we infer the presence of euechinoids about 268 million years ago. It would, from first principles, you can posit, as we have in a couple of papers, both Erkenbrack and Davidson and Thompson et al., that maybe that's when the double negative gate first evolved, when we have the difference between euechinoids when they diverge at that time. And so these two recent papers, that's what we implied. The question that we decided to further investigate, and this is where the new stuff I'm going to be telling you about, is can we say this with the data that we have available? How are there other ways that we might support that hypothesis? And the first thing to do is we need to know the timing of echinoid evolution, and we have the fossil record, but we can also use molecular data. And using 3 genes, we, in our first step of our analysis, produced a phylogeny for the crown group echinoids, the modern echinoids that we have. And we then, from that, we want to start to say, using, kind of putting this deep time perspective, when did the double negative gate evolve? Was it at that divergence? Or using, in this case, molecular data, because we don't have embryo or larval fossil record, but we do have a lot of molecular data from modern echinoids. How might we utilize approaches to understand this question? And the first thing that we wanted to do is to take a look at the modern echinoids and where do we have the double negative gate, and sort of map that on this phylogeny. And we know that the purple sea urchin, of course, we've just been talking about it, it's got the double negative gate. A closely allied euechinoid, Litocynus variegatus, also is thought to have the double negative gate. Whoops. Well, somehow I went, yep, thanks. Now, let's move up. And I should say that purple sea urchin and Litocynus are a part of a larger group of regular euechinoids. Let's move into the irregular euechinoids. And Echinocardium chordatum, which has been studied by Yamazaki and others. And there they also find evidence for the double negative gate and the expression we see here for PMAR in the micromeres. They call it micro 1. PMAR 1, micro 1 are the same thing. And other expression patterns also indicate that this irregular echinoid has the double negative gate. Here's Hess-C, so the rest of the embryo Hess-C is expressed, but not in the micromeres. Same thing for ALX. And here we see a summary. So the double negative gate exists also in this irregular echinoid. Okay, let's now go to another kind of irregular echinoid, a sand dollar, Scaphochinus mirabilis. We don't have a genome for that, so we've indicated its presence. It's a member of the Scutelidae, and these are the closely related genera for Scaphochinus, but that's in our phylogenetic tree where it exists. And also Yamazaki and others have studied this, and the expression patterns for Hess-C and PMAR or micro-1 and ALX all indicate that the double negative gate is in existence in this irregular echinoid. So we can put this on our tree over here. Now, if we go outside of the euechinoids, we know the two sideroids that have been looked at, Eusidaris and Prionosidaris, don't have the double negative gate. And this has also been examined in other classes of crown group echinoids, sea stars and ophiuroids. We have some examples where the double negative gate does not exist. So how can we quantitatively map the evolution of the double negative gate using this data that we have currently? And we can go about this using an approach called ancestral state reconstruction. It's sort of within a Bayesian framework, and the data that we have is the presence or absence of the double negative gate, as I've just shown you, within the modern groups. And what we're going to see here is, through this analysis, these little pie charts are going to show us the probability that the double negative gate was present or absent in the last common ancestor at that node. So the group of regular euechinoids, which we'll call echinacea here, and irregular echinoids. There are some also other regular echinoids in the euechinoids up here. But this group, the probability that the double negative gate was present in the last common ancestor at this node is favored, 0.65 to 0.35. I should say blue is yes, is favored, and 0.35 is not. So the probability is that it was present. If we go back to the node from this data, where the last common ancestor of the euechinoids is assessed, the probability also is that the double negative gate was present at the last common ancestor of euechinoids. When we get to the last common ancestor of this analysis, which we'll call echinoidea, but this is for all modern echinoids, and in fact, we know from the fossil record the last common ancestor is something called Archaeosidaris. This analysis does not tell us the answer. It maybe not, but it's not definitive. And also, if we look at the last common ancestor of the sideroids, we can see that it pretty strongly says that there was not a double negative gate present at this time. Okay. I think I have the double negative gate clicker here, don't I? So, so we've done this ancestral state reconstruction, and the theme has been let's try and place these things within time, and we're using molecular data from the modern organisms here, not the fossil record. So, well, we are going to use the fossil record, but we're going to combine that with this. And so we're going to do a molecular divergence dating with a fossil calibrated molecular clock, and within a Bayesian framework where we calculate this with likelihood from the sequence data, priors for rates of evolution, and the divergence times are calculated from fossils. And the time tree that we've produced is from a bunch of fossils where we've been able to calibrate 6 nodes. And these are some of the fossils that we've used. And this is hard to read, but basically the time tree that's produced, the blue or purple bars are the range of times that these nodes may have evolved. And we can throw that together with the pie charts of the ancestral state reconstruction and say that, for instance, the last common ancestor of euechinoids, whoops, try and go backwards here, was between 234 and 254 million years ago, and it was likely had the double negative gate. Now, we get a very young time in the time duration for the sideroids, but that's because we haven't calibrated the sideroids with fossils, that sideroid node. But we think that this is an ongoing analysis, and we think that it's going to be pretty powerful and interesting to understand the evolution of gene regulatory networks within deep time. So, sort of summing up here, using this paleogenomic approach, we can determine ancestral gene regulatory networks and constrain when those evolved. Our preliminary results that I've shown you here is that it's double negative gate is likely not present in ancestral sideroids, but it is likely present in ancestral euechinoids. We can't say with great confidence that it's traceable to the euechinoid-sideroid divergence, but that's also based on adult fossil specimens, and this is the larval embryological data that we're using. So that's part of the interesting part of this analysis. Paleogenomics offers we think the future is a rigorous quantitative framework for tracing the timeline of gene regulatory network evolution. And we can start to put some times and some geological dates on when these things that we have determined in the lab first appeared. Future work, we're currently working with Andy Cameron and others on a diadematoid, which is this representative of this other group of euechinoids, Centrostephanus, to establish the presence of the double negative gate, which we think will further strengthen our analysis. And of course, additional data would only make this better and better. I would, of course, like to thank Eric Davidson. In collaboration with him, we had a grant from NSF, which is winding down, at NSF called Creative, where you could get, if you got 2 different directorates of NSF, in this case we got biology and geology, to agree to put money up. We always found it, in doing the things that we did, hard for NSF to throw money at some paleo and molecular biology study, because they only understood half of it, depending upon where you went. I'd also like to thank Feng Gao, who was another part of the group of our creative efforts. And we'd like to dedicate this work to memory of Eric Davidson. Here is a picture that was taken in 1998 in China. I think I know who might have taken this picture. But this is Eric with, here's Feng Gao, by the way. And these are some members of our one of our Chinese collaborators' family, of Zhang Yuan Chen, and somebody who worked in his lab. But we have, I guess, a memory of a very vigorous Eric Davidson. Thank you very much.